Thanks very much to the organizers for a really interesting meeting, and thanks to all of you for staying for the last 3 talks. Of course, these are going to be the best 3 talks of the conference, so we'll give you a reason to have stayed. My disclosures are on the first slide. So I'm interested in metabolism. So let's talk about how things work. You know, if you got here an electric car, or you plugged in an electric toaster or teapot today, you depended upon high energy electrons, right? So in a vehicle like this, there's a huge battery, and there's high energy electrons that go to all the moving parts. The same thing in a toaster, even though it's plugged into a wall, there's high energy electrons that's going to the heating coil. Now, what you might not realize is that there's a flow of high energy electrons in all living things. So really, the system starts with the sun and distribution of photons that land on planet Earth and grow plant matter, right? And so that's living stuff. And then the animals come along, and the animals eat the plants. They eat the protein, fat, and carbohydrate in the plant matter, and they grow entire animals. Then we come along, and we either eat the plants directly, or we eat the animals that eat the plants. And from that biomass that we're taking, whose energy came from the sun, we run metabolism, right? And so what does that mean? It means that we convert everything that we eat into everything that we are and everything that we do through thousands of different reactions. Nearly all the work in biology is done with the energy delta, the energy difference between the energy level of the electrons that are captured by NAD+, and then the lower level of the energy of electrons that are joined with oxygen as the ultimate electron acceptor forming water. And if you don't know what I mean, just take a look at this diagram over here that is kind of an illustration of all the power in life, is that the energy of the electrons coming in is like this energy that is captured by energy acceptors, lower and lower energy acceptors like coenzyme Q, iron sulfur clusters, cytochrome oxidase, etc. And that delta between the energy of the electrons of NADH and the energy of electrons joining water can actually turn the crank and do work. Just like that water mill can grind rice into rice flour. That's really the way things work. And the mediators of these high energy electrons are 4 NAD coenzymes, not 1 NAD coenzyme, but 4. NAD plus on the left, NADH on the right. You can see that there's a typical substrate. This could be malate to oxaloacetate here, an alcohol to a ketone in this case. And what's happening here is high energy electrons, 2 of them, as a hydride, are captured on this NAD forming NADH. And so that's used to capture electrons from our food, so when we're oxidizing protein, fat, and carbohydrate. And then at the intermitochondrial membrane, NADH donates the electrons right into the electron transfer chain in order to make ATP, and then also to maintain blood glucose and to do ketogenesis, right? And to generate ketone bodies. Same electrons are powering that process. The electrons that were captured on NAD are donated by NADH. Now, this 2-oxygen of NAD can become NADP. No one's talked about the entire weekend, but NADP captures the electrons in the pentose phosphate pathway, right? Because those fields of corn out in Iowa or fields of rice, a lot of carbohydrate, right? That's a hexose. Glucose metabolism is, we run our body on glucose metabolism, but we run the information systems of DNA and RNA on 5-carbon metabolism. And there's no chemist of any skill anywhere in the world that can precisely take D-glucose and make ribose 5-phosphate, but every single bacterium and every single cell in your body knows how to run the pentose phosphate pathway using NADP to specifically remove one carbon from glucose to generate ribose 5-phosphate for all of the RNA and DNA precursors in your body. And then, of course, the NADPH is now an electron donor. And NADPH donates the electrons in the detoxification of free radical species, so in reactivating glutathione. We heard a lot about ROS generation, but you might not have realized that NADPH is required to detoxify ROS, as well as to make lipids and steroid hormones that are so important for life. And so we got interested in NAD biosynthesis a number of years ago. It's been said by others that there's something called a de novo pathway. It actually requires oxygen from tryptophan to go in 8 steps to make NAD. And then it was discovered again by other people that nicotinamide is a very important precursor. It's got the pyridine ring, and it just takes 2 steps to go from nicotinamide to NAD. And then nicotinamide can be degraded. So nicotinamide is a broken down piece of NAD. Nicotinamide can be further degraded all the way down to nicotinic acid, and then that can make NAD. And so we found ourselves working on an amazing enzyme here called glutamine-dependent NAD synthetase. Nobody really knew how that worked, where the nitrogen, the final nitrogen group in NAD came from. And we recognized how that was done, and so I think it was around 20 years ago, 2003, that we discovered the eukaryotic NAD synthetase. We called it QNS1 in yeast and humans, and how that enzyme worked. And then in the course of doing that work, we started questioning the received wisdom of how NAD is made and whether people understood all the pathways. We knocked out the NAD synthetase. There's not supposed to be another way to make NAD in yeast if you knock that out. But in the course of doing that, we discovered the NR pathway, right? So we discovered that NR is a previously unanticipated vitamin precursor of NAD in everything from yeast all the way to humans, nicotinamide riboside kinase in yeast, and then nicotinamide riboside kinase 1 and 2 in humans. So that was a nice little thing. So, and so now we know how NAD is made pretty much. And now I want to say something about these 4 NAD turnover pathways that we've discussed, because I think that there's been a sort of an unfair emphasis and unfair understanding of them. So monoADPribosylation is a strange term because it's catalyzed by members of the PARP, polyADPribosylation family, but it's monoADPribosylation, which basically just means that an ADP ribose unit is taken from NAD and forms a protein post-translational modification. But the thing that's really interesting about this is that in coronavirus infection, when there's double-stranded RNA inside of our eukaryotic cells, we can recognize that as a foreign body without an antibody response. This is innate immune response. And we showed actually during the coronavirus pandemic that there's 5 members of the PARP superfamily that are induced by coronavirus. Look at this, PARP 7, 14, 13, PARP 9, PARP 10. These are PARPs that literally no one ever talked about. 5 of them at least are induced by coronavirus infection and the innate immune response. So these are profoundly induced, highly induced genes. 5 or 6 different members of the monoADPribosylation members of the PARP superfamily. Then PARP, of course, we've talked about a number of times during the meeting. That's when an enzyme called PARP1 or PARP2 keeps on taking NAD and basically adding it to a polymer of ADP ribose. It can be stuck to a protein. It can be free in the cytoplasm. And again, this is a highly, highly induced process. So it requires DNA damage or reactive oxygen species to initiate this, and then suddenly PARP starts degrading NAD and making all of these signals that do tremendous amounts of things. So we have monoADPribosylation, I showed you, 20-fold induced. PARP is 20 at least fold induced. And then cyclic ADP-ribose formation. Not sure if I have a slide for that, but it's done by CD38 mainly, and by SARM1. There's two different families, and these are highly, highly induced. So cyclic ADP-ribose formation and NADP formation is 100 or 1,000-fold induced. SARM1 gets activated by intracellular NMN. Intracellular NMN in neurons is a sign of neuronal damage and activates this enzyme SARM1. So these are highly, highly induced. And then I still haven't mentioned the other NAD turnover reaction, the most famous one, Sirtuins. And the remarkable thing about Sirtuins is sort of how unregulated they are. So for example, they were said to be so smart in mitochondria, Sirt3, that it's going to activate a whole bunch of enzymes, but Sirt3 doesn't decide where the acetyl group is going to be on any lysine. That actually just gets put on by mass action. So in mass action, particular lysine residues that have a depressed pKa value get acetylated, and then Sirt3 just comes along like a garbage man and removes them. So I think the point of this slide is to show that MARylation is highly induced by infection and is druggable. PARylation is highly induced by DNA damage. SARM1 is highly induced by NMN. And then the Sirtuin investment thesis, the idea that Sirtuins are the key mediators of NAD has been intensively studied and has actually been one of the biggest drains on biomedical research in history and has almost nothing to show specifically in aging biology. We've seen some things about Sirt6 at this meeting, but overall Sirtuins are not longevity genes. They're not conserved as longevity genes. And we could see that from an earlier talk today when it was emphasized how they benefit the oldest mother cells. That it's only benefiting a mother cell that's about 21 generations old. So that's literally 1 in 5 million cells. And half of the cells in a culture have never even been a mother. You could regenerate a yeast culture with any one cell, and its Sir2 is only benefiting 1 out of 5 million cells. And then when Walter Longo at USC did a different type of aging assay called chronological aging, look at what he found. He found that Sir2 deletion as well as calorie restriction both extend lifespan. So it's actually bad to have Sir2 to live a long time in the yeast chronological aging assay. And this is actually relevant to yeast biology because we've been carrying yeast around in our clay pots and making bread and beer and wine for thousands of years. So this tells us that yeast does not have a Sir2 gene for longevity because it actually does better in survival without the Sir2 gene. But I think that Sir2 gene was conserved for regulation of mating type gene expression and not for longevity, which is really a problem in the field because it's so widely believed that Sir2 and Sirtuins are longevity genes. So I just told you that it shortens the lifespan of every cell in chronological aging, that it was reported initially in worms and then in flies in the early 2000s to be a longevity gene in animals. That would have been amazing. David Gems wrote an editorial about that, but then in 2011, David Gems and Linda Partridge and people from 8 or 9 different institutions showed that these data were not reproducible. And in fact, loss of 1 copy of the Sir2 gene in flies extends lifespan. And 4 groups did whole body overexpression, of SIRT1 with no lifespan extension, and then one group obtained 2 transgenic lines in the brain with SIRT1 overexpressed in the brain. One of those transgenic lines did not extend lifespan. The other thing seemed to, presumably due to a position effect, but amazingly, the group was able to conclude that SIRT1 overexpression in the brain does extend lifespan, even though in their own hands it was not a reproducible result. So this is really, really a problem. And SIRT6, we have seen some data at this meeting that in a sex and to some degree strain-specific manner extends lifespan in mice, but at the cost of higher blood sugar, which is not likely to work in people. And so there's inaccurate information in hundreds of review articles, each with hundreds to thousands of citations. Thousands of research projects are mispremised and suffer consequent framing bias, confirmation bias, hype, and harking, that's hypothesis, after the results are known, with billions of dollars misallocated. The resveratrol story is wrong and as constructed, non-falsifiable. Derek Lowe recently wrote in a Science blog article that articles actually by Bauer and resveratrol, the most highly cited papers in longevity, are wrong and cannot be right. The implications for popular expectations of longevity are enormous. Why? Because if it were true that sirtuins are dominantly acting, positively acting longevity genes, then we would all want more copies of them, and CRISPR would change everything. But as we've heard from the folks in the front row, aging in animals is actually highly polygenic, and there's not going to be one gene that there's no identified gene in mammals that you want more copies of. And so there's a huge bias that sirtuins mediate the effects of NAD modulation that is not supported by evidence. What is the basis for the function of the NAD system? As I showed you, high energy electrons that make our bodies function, that generate everything in our bodies, all the lipids, all the cell membrane, all the reactive oxygen species detoxification, etc. This is just one or two examples. Joe Bauer actually gave you a peek of this. This is from our lab, Digay et al. This is a mouse model of heart failure, and what you can see is that in the development of the mouse heart failure, and this is recapitulated in humans, there's super high expression of nicotinamide riboside kinase 2 with declining expression of NAD. There's also low NAD, low cardiac NAD, because of the declining expression of the NAMT pathway and the jacked up expression of the nicotinamide riboside kinase pathway. You can't restore NAD, cardiac NAD with nicotinamide, but you can with oral nicotinamide riboside. And this preserves the ejection fraction of these mice. And this has led to multiple registered clinical trials of nicotinamide riboside in the context of heart failure. So you can see here's the mouse result. Left ventricular ejection fraction is better, fraction shortening better, everything that we measured about these mice were better. Their NAD was under attack. Now, here's another thing that was amazing that our lab did, is that in a postpartum mouse, so a new mother mouse and a new mother rat, not only does their liver circulate protein, fat, and carbohydrates in the mammary, right? A new mother is making milk for the babies, right? That's how life works. But the amazing thing that we discovered is that a new mother, a lactating control mouse, has lower liver NAD. And she's circulating a high level of blood NAD. When you supplement the new mother mouse, you can support her hepatic NAD system, and you can super induce the circulation of a plasma NAD metabolome. And the amazing thing is that NAD is going to the mammary. So the lactating mouse has a 20 to 30-fold increased level of NAD that is being supported by the new mother's liver. And so when we did this work, The amazing thing is the offspring benefited tremendously. So the mouse on the top is a normal mouse whose mother was given normal chow. This mouse on the bottom is the offspring of an NR supplemented mouse. And if you look at the tails, they can get across this balance beam without any training, without use of their tail. And so there were benefits to the offspring of NR supplemented mice that are in this paper that has been published, and I encourage you all to look it up. It was rather a remarkable story. But essentially the theme of 20 years of research in our laboratory is that there are conditions that challenge the NAD system. The emojis didn't work perfectly here, but here are some of the key things, like alcohol metabolism damages the hepatic, the liver NAD system in human beings. Overnutrition, that means eating too much. Noise sufficient to produce hearing loss. Sun and oxygen damage by activating the PARP system. Changing time zones in irregular hours disturb the NAD system. Infection and inflammation. I showed you some of those data on coronavirus. Disturb the NAD system. Neurodegeneration, mitochondrial disease in humans, and heart failure disturb the NAD system. Pregnancy and postpartum, and then finally aging as well, there's a decline in the NAD system. So it's in the systems in which NAD is under attack that NR is going to be most important. Happy to say that we've been able to develop this safely. This is just one of our many safety trials. This shows dose-dependent increases in nicotinamide riboside without adverse events. So what are some of the indications for NAD? And we basically, we find that the metabolomics and the gene expression is our guide. So we see NADPH come under attack, like in mouse models of overnutrition and diabetes. If we see NAD come under attack, like in noise-induced hearing loss or in sun damage, and we see the nicotinamide riboside kinase genes are induced, there's a very good likelihood that a clinical trial in that area could be positive, but you have to run it for sufficient time. You have to have the right patient population. So heart failure, inflammation. There are now 7 completed, replicated human studies that show anti-inflammatory effects of nicotinamide riboside. Am I going to tell you that nicotinamide riboside is going to extend human lifespan? No, I don't think anything is going to extend human lifespan. But I'm telling you that I think there's very good reason to think that NR supports resiliency and repair processes that can allow us to age better, and particularly in many of the conditions of metabolic stress in which the NAD system is under attack. We decided in a new proposal to the National Institutes on Aging to look at the rate of skin healing as a function of human age. So we know that people that are younger can heal from scratches and scrapes and burns very rapidly, much more rapidly than older people. Skin is the largest tissue in the human body, right, because of the surface area to volume of a human being. And so what we're doing is we're working with a dermatologist at City of Hope who's removing non-malignant skin lesions, right, of a particular size from shave excisions, and we're looking at the rate of that healing as a function of age, as a function of the baseline NAD status, and then in a subsequent trial, we're looking to see if we could boost the NAD system and accelerate the repair of these wounds, and particularly in older people. So the conclusions that I brought you this weekend are that there are 4 NAD coenzymes that are the central catalysts of metabolism. I don't want you to ever forget about NADP and NADPH. 3 of the 4 NAD-consuming pathways are intensely regulated, while one less regulated system, the Sirtuin system, has been intensely hyped with very little to show. And there's very little reason to think that Sirtuin modulation is the primary mediator of NAD repletion approaches. Quantitative targeted NAD metabolomics allowed us to discover many conditions in which the NAD system comes under attack. The nicotinamide riboside kinase pathway is both constitutive, that's mostly NRK1, and inducible, that's mostly nicotinamide riboside kinase 2, and is a biological signal that a tissue is under attack for its NAD system and is looking for NR in order to respond to it and potentially promote its healthy metabolism. I'd be more than happy to take your questions. Thank you.
感谢 Brenner 教授。请为这位白色衣上衣的女士传递话筒。
Thank you. Terrific talk, as usual. I'd like to ask why you took skin as your main target, because I mean, in the scientific field, when we talk about the NAD, we always think about like something related to mitochondria and then something related to muscle, whatever. But skin seems not something that is into that.
That's because you haven't thought enough about skin. I never do it integrating. If most people are doing something else, then I don't want to do it. I mean, I'm very interested in liver. I'm also very interested in brain health. But you know, skin, we actually know that you know, lipid biosynthesis, nucleotide biosynthesis, skin repair, all depends strongly on the NAD system. We know from mouse models that there's slower healing in older mice. The first human data on the NAD system in which people claimed that NAD declines in aging, that was done with skin. So I think that skin is an outstanding way. It's an accessible tissue. We can take it at a cancer center from, you know, non-malignant patients. So I think it's going to be a great system. There's also topical interest in nicotinamide riboside in the skin. In fact, I'm going to Seoul, South Korea tomorrow morning, and we have a product in which NR is actually topically available to skin, and it works.
请给王教授传递话筒。
Yeah.
So I just have a comment and a very interesting observation of your data. I saw that the COVID virus, coronavirus infection caused PARP upregulation.
Yeah.
Also, NAD is inside of it.
Yeah.
It's like NAD is a stress responsive gene.
Yeah, so in most of the data sets, you could see 3 NAD biosynthetic genes that were increasing along with the PARP consumers, right? Those 3 genes were NAMPT, nicotinamide riboside kinase 1, and nicotinamide riboside kinase 2. Now, one of the problems with NRK2 is that you have to do really good RNA-seq to see whether you're getting substantive NRK2 expression, because the fold change NRK2 can be very, very impressive because the level is so low in a lot of tissues. But it's clear that what's called the amidated pathway of NAMPT and nicotinamide riboside kinase both tend to come up when you get this double-stranded RNA, the MAV1 system that is activating type 1 interferons. And so it looks like, and the nice thing is, there are a number of completed and in-process clinical trials of COVID recovery. There isn't one yet that's completed where it was NR alone, but NR in combination with 3 other over-the-counter things actually succeeded in a phase 2/3 at accelerating time to recovery from COVID. So we think that this is an important axis of metabolic resiliency.
Hello, Professor. I have some questions, and I like for your advice. As you said, Sirtuin is not available for a biomarker of inflammatory agent, but my brain T-cell sequence results showed in Sirtuin, and my experience infectious mice had a connective dysfunction. Can you give me some suggestion? Thank you.
It would help me if you repeated that a little bit. Something about sirtuins.
My experience in infectious mice has some cognitive dysfunction, and my brain T cell sequence result shows a certain expression.
Okay, so here's the problem with this, most of the sirtuin, nearly all of the sirtuin papers, is that if you simply look for correlative biomarkers of SIRT1 or SIRT3, you'll find them with NR supplementation. So for example, in Kanto's 20